Hey everybody, welcome to video 63. Today we're going to be talking about the most important part of the urinary system, and that is the nephron. Everything the kidney is able to do when it comes to filtering our blood is because the kidney is packed with millions of these nephrons that all take advantage of some certain things about physics in order to be able to do their job and to do it efficiently. So we're going to be looking at this at a very microscopic level. Recall that the function of the urinary system is to filter your blood. We went through those things that the kidney filters. It's also going to be responsible for resorbing water and salts. The urinary system will dispose of waste products and help maintain blood pressure. And we'll go over a few of those and how they're able to do that later. It's going to be responsible for stimulating the production of red blood cells. Or erythrocytes. So these four things, we're mainly going to be focusing on filtering blood, resorbing water, and salts, but all three of these are also very important. So the nephron is the structural and functional unit of the kidney. Each one is jam-packed full with millions of nephrons, and when we talk about the function of the kidney and its job, filtering blood, resorbing water, and salts, it all happens at the nephron level. The nephron is going to consist of the glomerulus, Bowman's capsule, the proximal convoluted tubule, which you'll see abbreviated PCT, the loop of Henle, and the distal convoluted tubule, which we'll see abbreviated the DCT. Here we can see a few of those all put together here with that glomerulus, Bowman's capsule here, the PCT, loop of Henle, DCT. We'll get into all of that going here. So the renal artery, as it is entering the kidney, is going to subdivide many times until eventually it will be known as an afferent arteriole. So it's still going towards arteriole here. That afferent arteriole will knot itself up in its capillary bed. So basically a capillary bed, but all knotted up, will be known as a glomerulus. And it's going to be so small that there actually will be little tiny filtration slits in between the cells of that afferent arteriole. Because of those little tiny spaces in between the cells, high blood pressure is going to force materials out through those filtration slits. Think you still have a quarter of your blood every minute still going through, but you have a much smaller canal that it's able to run through, and so that's going to increase the pressure that you have here, and that's going to force some materials out. Blood will then exit through the efferent arteriole, which can eventually become the renal vein as it exits the kidney. Bowman's capsule is a cup-shaped structure that is going to kind of envelope around the glomerulus. So if this is my glomerulus, sort of this tangled mass capillary and this capillary bed here, Bowman's capsule, usually we're going to draw it as a C shape, but really it's going to be a little more three-dimensional of a C shape, so more like a cup shape around that glomerulus to catch those materials coming out. Here you can see a little more of that cup shape, that C shape here. When we talk about it being a C shape, that's just because we're looking at it in two dimensions. But there's the glomerulus, and there is the Bowman's capsule. Anything that is captured by the Bowman's capsule will move down through the rest of the nephron here in the proximal convoluted tubule. So the proximal convoluted tubule, loop of Henle, and distal convoluted tubule are going to sometimes extend into the medullary pyramid. Others are going to exist, while the other structures, glomerulus and Bowman's capsule, are going to exist in the renal cortex. Remember that cortex being the very thin outer membrane around, or I shouldn't say membrane, but outer layer of the kidney. But the function of the PCT, loop of Henle, and DCT are to create a lot of surface area and a lot of time and option for capillaries that are wrapped around the loop of Henle to be able to reabsorb water and salts that otherwise would end up in your urine. So here you can see that afferent arteriole and the efferent arteriole, afferent coming in, efferent leaving. After that efferent arteriole leaves, it will then go back and wrap itself around the distal convoluted tubule and then the loop of Henle. And then the proximal convoluted tubule, loop of Henle, and the distal convoluted tubule, because it will push out all sorts of materials here But you don't want all of those materials to end up in your urine. In fact, actually you want to end up resorbing most of them. And so it pushes them all out, but then comes back and is going to be reabsorbing a lot of the stuff that it wants back. So nephrons are highly efficient because they take advantage of osmotic gradients when they are reabsorbing water and salt. And they're going to do this through what's known as a countercurrent exchange, and we'll take a look at it in the loop of Henle. It's not a really easy concept to understand right away, so it's a little abstract, so hopefully I'm able to describe it here. But directions, or wastes are going to flow in the opposite direction as blood. So in the loop of Henle, you're going to have a loop of Henle and waste flowing in a certain direction, while blood is going to be flowing in the opposite direction. Remember those capillary beds wrapped around that loop of Henle, blood is going to be flowing in the opposite direction, and they're going to take advantage of diffusion here. So if we're looking at this from a loop of Henle standpoint here, you have, this is your loop of Henle, and you can see it's flowing from left to right, while your blood is flowing from right to left. Here, the loop of Henle has a high concentration of water compared to this area of your capillary, of that blood vessel here. And so water wants to move from areas of high water concentration to areas of low concentration. And so that is going to cause water to move out of the loop of Henle and into your bloodstream. Continue over here, you have all this water getting pulled out, and as a result, salt, sodium and chlorine and potassium, things like that, become a lot more concentrated because there's less water in that waste. And by the time you get over here, you have this part of the bloodstream here has a very low concentration of salt compared to the loop of Henle here. And so salts want to move from areas of high concentration to areas of low concentration. That's just what diffusion is. And so salt is going to move out of the loop of Henle and into your bloodstream. If we look at that from the bloodstream standpoint here, blood starts here. It has salt moving into it, and we just described why. There's a lot more salt in the loop of Henle in this area than compared to your bloodstream, and so salt is going to move in. And so that makes your blood super, super salty by the time it gets around to here, and so it has lower percentage of water compared to the loop of Henle right here, and so water is going to move into your blood to try to balance out that saltiness and try to get equal percentages of water between the two. So hopefully I described that okay. And so nephrons are going to form urine through three processes. They're going to filter things out, so those water and solutes being forced out of the glomerulus and into the Bowman's capsule because of high blood pressure. As a side note here, low blood pressure can be a very dangerous thing in humans. Things like massive blood loss, which is what a hemorrhage is, can cause kidney failure because you no longer have the high blood pressure required to push those wastes out of your blood. And as a result, you get kidney failure, they're not doing your job, and you get accumulation of waste in your blood. But then water and salts are going to be reabsorbed. Other things as well, things like glucose, are also going to be put back into your bloodstream. They were forced out, but they're going to get pushed back in. And in fact, 99% of the material that gets pushed out is going to get reabsorbed. So all that water, glucose, salts, and things like that are going to get reabsorbed. So approximately 180 liters of material get pushed out of your blood, but 99% of them are going to get pushed back into your blood. And then you will get secretion as well. Things like ammonia, hydrogen, potassium ions can be actually pulled out of your blood and put into the distal convoluted tubule. Maybe not enough of them got pushed out of the filtrate. Maybe they didn't get pushed out in the first place, or you still have too much. And so your body actually has active transport mechanisms where they're spending ATP to actually pull them out of your blood and push them into the distal convoluted tubule so that it can end up in the final urine product. So that is a look at the nephron, the most important part of our kidney. Your kidneys are made of millions of them, and without the nephrons taking advantage of diffusion, you'd end up with very, very dirty blood. And so we can thank nephrons for doing their job and keeping our blood